In this introduction, we'll talk about the details of the bioinformatics and data science behind variant detection. Our ultimate goal is to see how our coronavirus sample is different from the original coronavirus. We are especially curious if our sample belongs to the Delta variant group. We will be using the same techniques used by scientists to detect variants. Except where otherwise indicated, this content is available for use under CC BY 4.0. Our main objectives for this set of slides are to first review the important lecture content. Second, we will review big steps in the analysis, plus information that will be important for answering the questions in this activity. Third, we will cover the setup steps on AnVIL in detail. Let's review first. Genetic variants are simply a small change in DNA. These occur in the genome of an organism. Genetic variants create a lot of the diversity we see in the natural world, such as disease risk or your appearance. SARS coronavirus 2 has a genetic variant called the Delta variant. This variant surged during the fall of 2021. Changes in the Delta variant make it more infectious, while causing greater disease severity among people. This is because the Delta variants all have a consistent change in the genome where it codes for the spike protein. The spike protein is what allows coronavirus to invade human cells. Scientists were able to figure out that some small genetic changes resulted in a differently shaped spike protein. This is because of the central dogma linking DNA, RNA, and protein. Although the genetic material for coronavirus is actually RNA, the same principle applies. Affordable DNA sequencing helped researchers catch the genetic differences in coronavirus quickly. The price to sequence a genome has dropped even more than expected given the technological improvements. Today, researchers can sequence a whole genome for about $1,000. Part of the reason genomic sequencing has become so affordable is sequencing by synthesis. This technology is still commonly used today. Improvements are also constantly being made to this technology. For example, the Illumina company makes a machine called the NovaSeq that produces 20 billion sequences on one sample. That's equivalent to 1,000 human genomes. In this activity, we will be using sequence data that came from a NovaSeq machine. Other researchers already took care of the laboratory part of the research. This means that scientists already performed extraction of the RNA out of the virus, turned the RNA into DNA, and put the DNA in the sequencer. Right now, we have thousands of sequence fragments from throughout the sample's genome contained in these two FASTQ files. Before we do anything else, we want to make sure the raw data that we are using from the sequencer is good quality. The FastQC tool will help us check the data. You want to pay special attention to the basic statistics. Here you can see how many sequences have been flagged as poor quality. You also want to pay attention to the per-base sequence quality. If you see box plots in the green, that means quality is good. However, if you see box plots in the orange or red, that could mean there is a quality problem that needs to be addressed before proceeding. FastQC will use the quality scores from each read to help us determine if any data is low quality. It helps us feel confident that we can move forward with our analysis. Up next is the alignment. Alignments also help us compare the actual base pair sequences of our samples to the reference. This makes it easier to find mutations because they are compared side by side. In this example, the sample has a C in this position, while the reference genome has a T. This could mean that the sample is a variant. We will use the alignment tool BWAMEM to automate the alignment of our sample to the reference genome. Why do we want to do all of this in the first place? Overall, alignment helps us detect variation. It tells us if we've found a variant strain of coronavirus that looks different from the original virus. Coronavirus researchers look for differences in the genomes that could mean a new variant has emerged. In some cases, variants could be tied to different epidemiology. In our lab activity, we just want to see if there are genetic differences in the sample compared to the original SARS coronavirus genome. We can count that as a discovery. We will be using AnVIL as the platform to do our analyses. Everything is in the cloud, so there is no need to download anything. All calculations are performed by a powerful computer connected through the internet and don't need to happen on your computer. We will go over how to log into AnVIL in a moment, but first there are a few things that are important to pay attention to during this exercise. The first thing to remember is that in the raw data, each fragment of DNA that makes up a read has 4 lines associated with it. The first line is a unique identifier for that particular read. The second line is the sequence. The third line is a placeholder. Finally, the fourth line contains a quality score. When you run FastQC, look at the basic statistics and per base sequence quality sections of the report. We are looking for scores higher than 30 in order to feel comfortable moving on to the next step. Alignments help us place sequencing reads in context. It shows us where those bases can be found in the reference genome by comparing them side by side. Alignment tools like BWA-MEM figure out the optimal positioning of the reads next to the reference genome. They also minimize mismatches and gaps. In the lab exercise, we'll be using paired end reads. Paired data is made up of fragments that are read twice, forward and reverse. Paired data helps improve data accuracy. Finally, it's important to know about how SARS-CoV-2 variants have fluctuated over time. Here is some CDC data showing the abundance of different strains in late 2021 and early 2022. The infectious and severe disease-causing Delta variant was superabundant in November and early December of 2021. The Omicron variant became common afterward. The samples we are going to be using for this activity were collected in November 2021. While it's a good guess that these samples represent the Delta variant, it would be good to confirm this hypothesis with data. Now let's go back to the first part, getting set up on AnVIL. Let's go over how to log in to AnVIL first. First, go to the anvil.terra.bio website. Click on the hamburger menu on the top left. Then click Log In. In the pop-up window, make sure you click Sign in with Google. Microsoft capabilities are still under development. Now you can enter your Google account info. You can also click on Create Account if you haven't made your Google account yet. Be sure to tell the instructor your Google account name so the activity workspace can be shared with you. You should now see yourself logged in. Next we'll go over how to clone a workspace. This creates a copy of the important materials so that you can experiment in your own space without worrying about changing the original data. Click on the hamburger menu on the top left. Then click Workspaces. Look for a workspace called SARS-CoV-2 Genome. It should appear under My Workspaces, since it has already been shared with you. You can also go directly to the URL on the bottom of this slide. Click the teardrop button on the right-hand side of the workspace and then click Clone. Rename the copy to include your name. Your class should already be set up with a billing project. From the billing project drop-down, make sure you select the correct class. Check in with your instructor if you aren't sure about this. Then select Clone Workspace. Now that we have our workspace prepared, we will start working in Galaxy. In your new workspace, click on the Analyzes tab. Next, click on Start. You should see a pop-up window on the right-hand side. Click on the Galaxy logo to proceed. Click on Next. Then, click on Create to keep all settings as is. Your Galaxy environment is now provisioning. Click on the Galaxy icon. It will take 8 to 10 minutes to get your cloud computer set up. The environment is ready when you see the status change. Click the Open button to launch. Let's look at the Galaxy interface. Notice the 3 main sections on the Galaxy page: Tools, the main dashboard, and History. Tools, on the left side, includes all of the bioinformatics tool packages available for you to use. The main dashboard in the middle will be the main area for adjusting settings on the tools and viewing output. The History section, on the right side, will show all of the documents and files you've been using. Luckily, we linked to the original data when we cloned our workspace. Now we just need to bring it into Galaxy. Click on Upload Data in the Tools pane. Then click on Choose Remote Files at the bottom of the pop-up. Double-click the Workspace folder, then Tables, then Reference. Click on the FASTA file so that it is highlighted in green and click OK. Now that your reference has been added to the download list, click Choose Remote Files, again to add the 2 sample files. Double-click the Workspace folder, then Tables, then Samples. Click the 2 sample FASTQ files so that they are highlighted in green and click OK. Now you are ready to upload the 3 files. Click Start. Your files have uploaded successfully. You can click Close. Confirm your upload worked by looking at the file names in the History pane. They should show up in green with no errors. When you click the eye icon to preview the uploaded FASTQ files, they should look something like this. Next, we will check the quality of our data using the FastQC tool. Open the FastQ Quality Control menu. Next, click FastQC, Read Quality Reports, to check the quality of the sample. Make sure you select the Forward Reads Sample FastQ file from the first drop-down menu. Leave everything else as is and click Run Tool. Here we can see the FastQC job going from In Progress in yellow to Done in green. If you get an error, try running the job one more time. Click on the eye icon next to the web page result to view the FastQC report. Your results should look something like this. You will need to look at the basic statistics and per base sequence quality to answer a few questions. Now we are ready to perform the alignment step. Open Map with BWA-MEM under the Mapping menu. Select the Use a Genome from History and Build Index option from the first drop-down menu, because we want to supply the reference genome ourselves. Underneath, make sure the SARS-CoV-2 reference FASTA file has been selected. Once you have set the reference options, you need to tell the aligner which files belong to your sample. Make sure that you have selected Paired under the Single or Paired End Reads menu. Select your Forward Reads FastQ file as the first set of reads. Select your Reverse Reads FastQ as the second set of reads. Click Execute. Finally, click Run Tool. Once your alignment has completed successfully, you are ready to take a look at the genome. Scroll down in the Tools menu. Under Statistics and Visualization, expand the Graph, Display Data menu. Scroll down to find JBrowse Genome Browser. Then, click on Use a Genome from History on the first drop-down menu. Make sure the reference genome FASTA file has been selected underneath. Click Run Tool. Once JBrowse has finished, click the eye icon next to the analysis. This should bring up the genome viewer. You will need to use the magnifying glasses highlighted here to zoom in and see the individual base pairs. This doesn't tell you anything about the samples though. Let's pull up JBrowse again, but this time, we will add our alignment. Click on JBrowse in your Tools pane. Then click on Use a Genome from History on the first drop-down menu. Make sure the reference genome FASTA file has been selected underneath. A bit lower on the page, you will see an option for Track Group. Click the Insert Track Group button. Next, click the Insert Annotation Track button. There are a few options you want to specify to add the alignment. First, select BAM Pileups under the Track Type menu. Below that, select Yes under Auto-Generate SNP Track. Finally, add an additional 0 to the maximum size of BAM chunks and click Execute. Open your results as you did before. You will see an Available Tracks menu on the left side of the genome. Make sure both boxes are checked. To look at position 24410 in the genome, you can type this number into the search box. This will take you to a single nucleotide that is different in our sample compared to the reference. The highlighted color and the A's listed throughout our sample reads are a good way to spot this. You can determine the exact location by hovering over the position bar. Another example of a mutation can be found at position 23603. It's a good idea to export your history so that your collaborators can see what you did. Click on the History menu and click on Export History to File. Make sure you select To a Remote File. Then, click to select where to export your history. Select your workspace name, then select Other Data. Finally, select Files. Then click Select this folder. Make sure the export directory looks correct. Next, name your history, SARS Galaxy Variant Detection, and click Export. Back at your workspace, click on the Data tab, and the Files folder. You should now see the history export in your files. If you click on the file, you can download it or view it in Google Cloud Storage browser. Now that we've explored our sample, let's shut down our cloud computer. Make sure you have answered all the questions before moving on to this step. Back in AnVIL, click on the galaxy icon on the right. Click on Settings. Select Delete Environment. Select the Delete Everything, including Persistent Disk, option. Finally, click Delete. Great job! You have now completed the activity. Thank you to everyone who contributed.